So yeah, I'm zooming in from the Netherlands. So I am an assistant professor at the University of Groningen. And what I want to talk about today in more kind of like an introductory scheme is everything you need to take into account actually before you start cloning. And this is something I call the cloning strategy. So it's definitely part of the kind of like design phase of your project. So this is why we included it into our webinar about getting started, because I think it's really important. So, how do I proceed here? Yeah, so as a brief overview, so Ian just told you how you can basically condense your ideas. So you walk from a protein that you want to use or an enzyme that you want to use through all the different databases, and you extract the genetic information. And what I want to talk about now is how do you get this genetic information into kind of like your circuit that you want to implement, because usually you want to have it under a certain promoter. It's supposed to be inducible. It's maybe supposed to be linked to other functions such that you can actually build a molecular machine out of it. And how you do or how you build this circuit in such a way that you can actually feed it into this kind of like design, build, test, and improve cycle. Because what you're likely going to figure out is that your first design won't work so well. So you also need to take into account kind of like ways how you can rapidly improve your design. And this is an essential consideration for something that I call the cloning strategy. So yeah, oops, let's go back. Yeah. So in summary, part of the design phase is to develop a strategy, how you can build a circuit that is easy to improve later on. So in my view, a cloning strategy needs three major considerations. So before I start, let's just quickly review what I consider cloning. So cloning is really the process of taking a piece of DNA, and that might come from a natural source. Maybe you PCR it out of the genome of your favorite bacteria, or you kind of extract the sequence from databases, and then you send it to a DNA synthesis company, and those people will deliver you your gene. And then you clone it into a backbone that is able to replicate in your favorite organism that you use to fulfill your function. So, and this is already kind of like the first consideration you need to take into account. So the first question is always, what host organism are you going to use? Because this determines what backbone or vector. So backbone or vector are kind of like interchangeable terms. So these are the components that actually make your genetic circuit replicatable in an organism. And I'm going to talk about this in a little bit more detail. So this is the first consideration. Are you going to work with bacteria, yeast, plants, mammalian cells? Because once you decided on one, it's actually not that straightforward to interchange these. So you kind of like need to decide that early on. And the second question is, how do you assemble your genes of interest into a circuit? And here you need to choose an assembly method, and I'm going to introduce the most useful ones to you, but we have special seminars on many of these assembly methods. So I'm going to point out at certain points during my talk when you actually should attend another webinar on those. And the third point is what I already mentioned, is the question, how are you going to troubleshoot the circuit performance? So you want to have a flexible and modular design and assembly strategy in place for both the backbone and the circuit, such that once you figure out your first design doesn't work, you can really rapidly kind of like re-engineer it, test it, because that's really essential for you, especially always in science, but especially in iGEM, because time and money is usually short. So you want to have that strategy in place. So altogether, this is kind of like all these three components I call the cloning strategy. So as I mentioned before, it is important to think of a cloning strategy during the design phase, because the cloning is really an essential component of the build phase, and this is really just to hammer it in. And cloning, having a strategy in place really enables the improvement phase, and the cloning strategy depends on the design and also on the test phase. So this is just reiterating what I told you before, but really have to put that on the slide. The points for consideration: what host organism are you going to work with? How complex is your circuit? So how many genes are going to be involved? How many plasmids are you going to need? Or are you even going to work on the chromosome? For the improvement phase, it's good to consider already in the design phase and also for the test phase, what data are you expecting? So what component of the circuit is actually testable? Because that one is also the component you want to have interchangeable, because that's the one you can iteratively improve. And then again, the question, how do you ensure modularity and flexibility to change the design rapidly? So your cloning strategy is always first developed and tested in silico. So this is like something Alexis has already mentioned, and Ian has also mentioned. If you extract your DNA sequence from all these databases, the first thing you do is that you paste it into a virtual cloning environment. And we have special webinars on the use of different ones: SnapGene, Benchling, and Genius. So these are the most widely used one. It's mostly personal preference what you want to use. And those are the ones where you can then actually assemble your circuit and test your cloning strategy. So please visit one of these webinars, or actually, yeah, one of these. It's not a webinar, it's actually a demo. So similar to what Ian just did before, just like really walking you through how we can use it and how you can, navigate through it to actually get a first trial on your cloning strategy. And now really walking you through the different kind of like components I had talked about. So the first question is really what host organism you're going to work with. And I'm going to focus on bacteria and yeast, but everything I say also applies to mammalian cells, plants, or non-conventional bacteria or yeast. But most iGEM teams usually work in E. coli or Saccharomyces cerevisiae. So what is important to consider here is really one, your circuit, and the other one is your backbone, or as I said before, the interchangeable terminology for it, the vector. So the circuit design obviously depends on the vector, because bacteria and mammalia and eukaryotic cells such as Saccharomyces cerevisiae do have different translational machinery. So you need to take that into account when implementing your promoters. But what is even like earlier, before you, design the circuit, you need to think about the backbone. So, that's maybe like obvious, but you also need to know where you get backbones from. So how do you actually start, implementing that? So the genes we said you can extract from the database, you can, get from a company, but where do you get your backbone, a vector from? Because those are the ones that actually make your circuits replicatable in your chassis. So they need to have an origin of replication that is compatible with your host organism, and they also need to have selection markers. And here I really want to tell you two great resources for these type of backbones. Those are called vector collections, and the first one is for bacteria, and it is the abbreviation is the PCVA collection, the Standard European Vector Architecture. And it is really a great resource, and you can actually request that from this website. So the whole collection has a website where you can actually also, just request the actual DNA. And it is a system which with interchangeable selection markers, origins of replication, and multiple cloning sites, such that when you have your circuit designed, you can easily clone and swap it into different vector backbones. And, as I will talk about in a minute, for example, change origin of replication or add certain components to it. So it's a really useful source to get you started when you work with bacteria. Something similar exists for yeast. It's called the PRS series of plasmids. It's similar interchangeable selection markers, origin of replications, and multiple cloning sites. They're actually not that nicely hosted all together on one side, but I kind of like just pulled up some iGEM numbers for you where you can find different of these vectors. And once you kind of like look at them and you understand the design, you will also be able to find more. But these ones are already kind of like, I think, useful. Another source for yeast is the so-called yeast molecular or modular cloning toolkit that was developed by the Düber Lab. And it is basically a modular way how you can just assemble these PRS plasmids by yourself. So it's a toolkit that is available from iGEM as well. Check out the number. and you can buy it via iGEM, and they have a really great tutorial on it, how to assemble it. So you can do that by yourself. So just to kind of like give you an example of why it is important to have these backbone collections. One is, for example, if you want to change copy number of a plasmid. So given you have designed your circuit and now you don't know exactly, you know, if you want to have it a low copy plasmid or like a high copy plasmid. So here you can really just, you know, design one circuit and then clone it into different vectors and see where it like works the best. It is also important to mention that the PCVA collection has different origins of replication which are compatible with each other. So that is something you need to take into account when working with bacteria, that some of these origins can't be used together. So this is also something that is nicely explained in the resources on this PCVA website. Another example about the importance of about the importance of really thinking ahead of time about having vectors or backbones in place that you can use for insertion of your circuit is if you want to add a component. So usually, you know, synthetic biology and iGEM is all about complexity. So let's think you start kind of like simple. You want to test your first design, and then in a separate experiment, you test your second design. And then eventually you want to put it all together. It's really nice if you had already both circuits you tested independently in vectors which are compatible with each other, meaning they have different selection markers and they have different origins of replication, that then later on you can just transform them together and bring them into the cell together. So this makes it kind of like really easy to scale in complexity. All right. Moving on to the second strategy question, which is basically assembly of circuits. And here I will, in actually the next slide, will give you some examples of really good DNA assembly methods. So basically, how do you stick different pieces of DNA which you have either PCR'd from an organism or you know, bought from a company? How do you actually physically stick them together? But before I do it, I will really talk about the importance of modularity for troubleshooting. So again, my example of the circuit I have here. So these are different genes and they kind of like interact with each other. So B, like gene B, for example, B and C repress this red gene here. So this is kind of like, you know, a genetic circuit that you designed and you tested and you find out that it doesn't work. And in that case, I hope for you that you kind of like thought about this circuit in terms of modules. So let's say this like first gene and its promoter and terminator is like the first module, module A, and then B, gene B with its promoter and terminator are module B, and so on and so forth. And if you did that and you kind of like cloned it in a proper way, namely that it's, for example, flanked by restriction enzymes, then you can rapidly exchange modules within that circuit and see during the troubleshooting phase, so within this test cycle, during the improvement phase, which component works the best. So of course you can also further kind of like abstract that and modularize it and even think about the promoter in here as an extra module and the gene as an extra module and the terminator an extra module. So what I want to, you know, just tell you is think as modular as possible in terms of your cloning strategy by flanking different parts of your circuit by, for example, with restriction sites or any other measure to make it modular, because that will allow you to rapidly kind of like make a second design where here in this case you replaced part B by B star and then test if it works. And maybe it does. So all of this is actually great because you don't need to redesign the full circuit, but you rather exchange components. That means also, again, in in the iGEM mindset, it's really quick and cheap to troubleshoot because you don't need to get new primers, you don't need to get new DNA, so you just exchange little parts. And again, during iGEM, you don't have a lot of time, so the better you plan your cloning ahead, the more time and money you save later on. And this doesn't only apply for iGEM, it applies for science in general, of course, always does. So now about different assembly methods. Here we really have webinars in place that you should visit. So I briefly going to touch upon the biobrick assembly method, Gibson assembly, and Golden Gate assembly. So all of them are modular. So all of them are great for, you know, a basic cloning strategy that you want to have for synthetic biology. They are, like Gibson assembly and Golden Gate are scarless. I'll tell you in a minute why. And Golden Gate is actually really scalable. So it comes with some extra work, but if you want to test a lot of different things, it has some advantages. But again, if you want to really, you know, understand perfectly what it is about, visit the webinar. Biobrick assembly is based on restriction enzyme cloning. So like each part is always similar to what I showed before, flanked by restriction enzymes that allows you to interchange the parts. Gibson assembly, but then you always have a scar because you used a restriction enzyme. So the restriction enzyme, of course, has a certain sequence that is, you know, that's something we call a scar in genetic engineering because you cannot freely design it. This is not the case for Gibson assembly because here exonucleases during the reaction create large overhangs that allow annealing, and then you have more accurate assemblies and also more freedom in design. During Golden Gate assembly, during Golden Gate assembly, you basically have a pre-cloning step, and then later on, based on type 2 restriction enzymes, you assemble circuits. The most important message for today is if you know, need to design something and you don't understand fully what the pros and cons are, and you really can't design, Gibson assembly is always a good choice for most cloning strategies. It might not always be the perfect and best choice, but it always gets you where you want. So that's always a good choice. So if you want to read deeper about something, read about Gibson assembly. The other ones are like, have pros and cons for very specific applications. And yeah, with this, I really want to, you know, close out and just leave you with the take-home message that a well-defined cloning strategy is really essential to run the full cycle of design, build, test, and improve, and make a device that works. And again, we have specialized seminars on the different cloning apps I talked about, SnapGene, Benchling, and Genius, and then also on the different assembly methods, Gibson assembly, Golden Gate, and the biobrick standards. Yep. So with this, I'm gonna stop sharing my screen. And yeah, if there's a question, please feel free to ask.
So Sonia, you had a question on, I'm just asking more question, like what does scarless mean, or what does really a scar mean in cloning?
Yeah, so a scar is really a short sequence that cannot be designed freely by the user. So a scar, for example, if you want to work with restriction enzymes, so the restriction enzyme have a defined sequence that eventually can't be changed. So if you want to, for example, have your gene flanked by restriction enzymes, then there is a defined 6 or 4 base pair sequence between your promoter and your gene that you can't change. So if it turns out that this one is like really important for the strengths of the promoter, you know, you need to work around this scar. With the Gibson assembly, you don't have that. Does that make sense?
All right. I hope we answered your question on scars. So with that, if there's any more questions. Okay, so we did have another question. It says, can you explain more about why Gibson strategy is better?
Why Gibson strategy is better? Basically, for what I just said before, Gibson assembly leaves full freedom of design to the user. So again, you can like seamless attach a promoter to a gene in a design as you wish, because it's, you know, you don't have any restrictions on the sequence. Also, it's like very efficient. So you can efficient in terms of piecing several genes together. So restriction enzyme cloning, usually if you want to, for example, clone 3 genes together, or like a transcriptional unit, a promoter, a gene, and a terminator, that will already be difficult. So you rather go one by one. With Gibson assembly, you can really nicely, in one reaction, assemble, let's say, 10 pieces together. So it's, yeah, efficient in terms of how many pieces you can clone together in a single run.
I also do want to say that NEB has it's New England Biolabs, which is one of our sponsors. They have a lot of further information on Gibson, on biobrick assembly, on really all the assembly techniques, in case you want to get a really detailed look at just what the advantages are of each assembly method, and really look further into just how you would order that and how they work a little bit further. There's also YouTube videos on that. But further on that, we are going to have separate webinars on just Gibson assembly. You're going to have a separate webinar on yeast assembly. We're going to have a separate webinar on molecular cloning, and we're going to have a separate webinar on Golden Gate assembly. So yes, NEB.
And in the different seminars, we will demonstrate how to do these with all the different softwares that we have mentioned.
Yeah.
So there's one that says, what are the drawbacks of Golden Gate assembly? One drawback that can definitely come up from a Golden Gate assembly is you do have to have a lot of restriction sites. So you do have to understand that when you are designing this, that you have to have those restriction sites, you have to have those restriction enzymes. It can get a little cost ineffective, but it can save you a lot of time inside the lab, because it's basically a one pot where you can assemble 10 gene fragments in one little tube, instead of things like biobrick where you have to assemble them part by part. But definitely look at NEB. They have tons and tons of information on these different assembly methods, and I've used them a lot when I'm analyzing certain things for a specific reason.
Sorry, by the way, my Zoom is kind of frozen, so this is why I'm still sharing my screen, in case you wonder.
It's no problem anyways. Do we have any further questions that you guys can think of? I'll wait a second to see if anybody has any further. If you guys don't have any further questions, you guys can definitely contact us at measurement@igem.org. You can request Sonia, Alexi, or I, or whoever can help you at whoever's first available. If you have further questions on one of these specific blocks inside this webinar, I suggest requesting one of the speakers. We do want to say thank you for attending this webinar. We can't do this without you, and we're grateful for you guys putting in all your efforts to making this another spectacular iGEM year, considering all the things that everyone has to deal with across the globe. But we're happy to be here for you guys, and we're happy that you guys are here for us. And we just want to extend the invitation to contact us further at measurement@igem.org, and tune into our further webinars later in the summer.